and we'll start the presentation. So I welcome you all to the final live TA session for the course Introduction to Developmental Biology. Today we'll be discussing week 12 lectures corresponding to Evolutionary Developmental Biology, and the course instructor is Professor Subramaniam. My name is Ashutosh and we'll solve sample questions for related to the quiz and the similar format and covering some of the concepts covered in lecture week 12. And the set of instructions for all the students will be same. Please turn off your mic and video chat for smooth conduction of this session. I'll keep posting 10 questions, 10 to 12 questions similar to what you face in your assignment. Then I'll ask you to answer these questions via the chat box. You can mention whatever option you feel is the correct option. I'll reveal my correct answer, what is the correct answer, and we'll understand a little bit a concept about why the correct answer is the correct answer. So that is the general instructions for all the students, and those who have missed earlier classes, you do not have to worry. The recordings are available on the YouTube, and the link is provided by the NPTEL to you, and you can also access these presentations. So without any further delay and with that much of introduction, let me share my screens and then we can move ahead with the questions. Yep. So here we are for practice assignment week 12. So we'll solve some questions in correspondence to week 12 lectures, and I welcome you all to this live DA session for the course Introduction to Developmental Biology. So the course is taught by Professor Subramaniam in IIT Madras, and my name is Ashutosh and I'll be the teaching assistant. I'm from IIT Kanpur. So I hope you can see my screen and hear me clearly. Just give me a yes or no in the chat box and we'll proceed. Good, very good. Okay, so the first question of this session, it's a match the following question. On the left hand side we had Roman numerals 1, 2, 3, 4, 5, and on the right we have A, B, C, D, E, and you have to tell me what is the correct match. Gervine alkaloid, corn gloom, allometry, pharyngula, Buchnera, whale maxilla, heterotypy, SHH, phylotypic stage, IBPA. And the options are 1E, 2A, 3C, 4D, 5B. Second option 1D, 2C, 3E, 4A, 5B. Third option 1E, 2C, 3E, 4B, 5A. And the last option 1D, 2E, 3A, 4C and 5B. So these are the options before you, and you have to match the correct column left to its correct match on the right. Now you have to tell me which of these option is correct. Let's take a minute and allow time for all the students to solve it. And in a minute or two, I'll reveal the correct answer. Okay, it's time now I reveal the correct answer. So the correct answer to this question is option 2, and Ajay is absolutely correct. Option 2 is the correct answer. Correct match the following. So we have gervine alkaloid matching with SHH, allometry matching with whale maxilla, Buchnera matching with IBPA, heterotypy matching with corn gloom, phylotypic stage, pharyngula. So these are the correct matched options from Roman numerals 1, 2, 3, 4, 5 and A, B, C, D, E. So let's learn about some of the matches here. So the first match, gervine alkaloid, it's a very it's most discussed in the developmental biology course. If you see, we have encountered this in earlier in the SHH signaling. Gervine alkaloid is derived from this plant, Veratrum nigrum, and gervine specific mechanism of action is inhibition of the SHH pathway. So gervine specific mechanism of action on the SHH pathway involves interfering with the processing of Gli proteins. So if you remember earlier during when we were in the mid lectures of the developmental biology, we learnt about different signaling such as BMP, Wnt, FCH, FGF and SHH. So in SHH signaling, the Gli proteins are the effector molecule. So Gli proteins are those molecules which can translocate inside the nucleus and turn on transcription of SHH responsive genes. So when SHH pathway is active, it leads to processing of Gli proteins, and Gli protein can move inside the nucleus, turn on certain genes responsive to SHH pathway. So what does gervine alkaloid do? It inhibits the processing of Gli proteins. So normally Gli proteins are regulated by complex process that involves their activation or inhibition by presence of SHH ligands. So I'm not going into the detail of the SHH pathway, but gervine alkaloid is a inhibitor of SHH pathway. Gervine is thought to interfere with the processing of Gli proteins, preventing them from properly being activated. And this can lead to dysregulation of target genes, disrupting normal development, tissue regeneration, and other processes which are controlled by the SHH pathway. So this is a naturally occurring alkaloid, and this is the example of a sheep suffering from cyclopia. Cyclopia is a mutant in SHH pathway, or due to inhibition of SHH pathway, the sheep now has unified eye field. So there are no two separate eyes, but it has one eye in the center of its head, which resembles a mythological monster called Cyclops. That's how the disease has been named, cyclopia. So what happened to these sheep? These sheep were accidentally grazing on these plants, Veratrum nigrum, which was rich in the gervine alkaloid. Thereby, these gervine alkaloid inhibited the SHH and impaired the development of the pups born from such sheep. So gervine alkaloid is matching the SHH pathway. It's okay, Ajay. See, in a column of match the following, if you see carefully, you just have to know two or three matches which are correctly, then you can spot the correct answer. It's perfectly fine. Even I used to answer like that. Okay, so next we move on to allometry. So this was the second option, the second match, allometry and whale maxilla. So what is allometry in whale maxilla? So it is a consequence of modularity associated with heterometry. Heterochrony is allometry changes which occur when different parts of the organism grow at different rates. So we have the same organization. If you see between a human and a whale, both are mammals, they have same organization of the skull, and these jaw bones are called maxilla and mandible. But if you see here, maxilla in the whales is very enlarged and it grows longitudinally. This is known as allometry. The structures, the bones are similar, but in the whales, the maxilla is bigger. Allometry occurs when different part of organism grow at different rates. So this has a different rate of growth than other bones of the skull. As animals develop, their shape changes, a result of differences in timing and duration of growth in events. So allometry is a combination of heterometry and heterochrony. Heterochrony is time of gene expression, and what is the duration of the gene expression, changes in that. Heterometry is amount of gene expression. So the amount of growth and the time of growth is different, and the duration of the growth is also different. So these modifications of whale maxilla due to allometry allows whale for its breathing. So the whale has to come up to the surface and breathe through its blowhole. So for that, the nasal blowhole and the maxilla are elongated. Indeed, morphological evolution, especially within a phylum, is due primarily to changes in body size and relative sizes of the body parts. So, if we closely see closely related species such as mammals, then the body plan and everything is same, but the body size and body parts are different due to differential growth. A dramatic example of allometry come from whale skulls. In the very young whale embryo, the nose is in the usual mammalian position. So if you see embryonic structures in the both whale and human, both of them the nose is at usual mammalian position. However, the enormous growth of maxilla and premaxilla pushes over the frontal bone and forces the nose to the top of the skull. So this is what I was talking about. The blowhole is actually the nose that we have in humans. Share the similar structure, but due to elongation of the maxilla, now the nasal pore or the blowhole is situated on the top of the head of the whale, so that it can surface over to the water and then breathe. This new position of the nose turns into blowhole, allowing whale to have a large, highly specialized jaw apparatus to breathe while swimming at the water surface. So this is a clear example of allometry. Let us speak of another. Yes, Ajay, you have something to ask? Yeah, yeah, yeah. Yes. Yeah, I think, yeah, I might have to look at this because I'm not really aware. But I think when they are deep divers, they go inside and dive deep while fishing and hunting. I think for a single gust of breathing and air, so for the breathing and air, they take in lot of oxygen. For that, they can roam around in the deep waters for some time. But to exhale, they have to come out to the surface. So time and again they have to surface on the water and blow through their blowhole. Also, I want to comment that I am not aware how do whales actually breathe. I have to might look about it. It will be fun. You can also find it on Google. But this is what I think. While breathing in, they breathe lots of air. Then that can keep them afloat and dive deep for a longer period of time because they have bigger lungs and capacity. And for exhaling out, they have to come and blow through their blowhole. That's what I think happens. Okay. And the third point that I wanted to discuss here is phylotypic stage matching with pharyngula. So, what is a phylotypic stage? In embryology, a phylotypic stage or a phylotypic period is a particular developmental stage during mid embryogenesis where embryos of related species within a phylum express highest degree of morphological and molecular resemblance. So I can call a stage a phylotypic stage when within a phylum, let's say within vertebrates, where a fish embryo, a chicken embryo, a whale embryo, and a human embryo will share the highest degree of morphological similarity and molecular similarity also. So this is the last point of similarity between all the embryos in a phyla, and after that we see divergence. After that, we see species specific structure. So when we say that all our embryos, all related embryos or all embryos in a phylum have same embryological features, so the maximum stage up to which they resemble each other, that is called a phylotypic stage. After that, the structures can start to vary. And pharyngula is such a stage when the four distinguishing features of vertebrates have developed, such as a notochord, dorsal hollow nerve cord, post-anal tail, and a series of paired branchial slits. So this is the stage, this is the phylotypic stage of vertebrates called pharyngula, where we have branchial slits or the pharynx, tail, post-anal tail, nerve cord, and a notochord. So these four structures resemble each other, and at that stage of the embryo, we say that this is a phylotypic stage, and pharyngula is such a phylotypic stage in vertebrates. Cool. I hope it is clear to the students, and we move on to the next question. Which of the following statements are true? So this is a true false statement. Which of the following statements are true? Mind you, there might be multiple answer correct. Option A, environmental induction of phenotypes is advantageous as the phenotypes are already present in a large portion of the population. Option B, leg development in insects is controlled by UBX ultrabithorax, which inhibits distal less gene in posterior segments through its C-terminal polyarginine stretch. This is the option B. Option C, pregnant yellow agouti mice, when fed a diet with low methyl donors, will produce dark-skinned fit progeny. And option D, notochord in vertebrates is an example of phyletic constraint. Okay, I have read out all the four options to you, and you guys have to tell me which of the following statements are true. Let's take a minute or two to allow all the students to answer, and then I'll reveal the correct answer. Okay, I think it's enough time for students to think about the answer. And I see Ajay has answered option 2, 3, and 4. Okay. And for other students, in interest of time, I'll reveal what is the correct answer. So the first statement which is true, is statement 1. Environmental induction of phenotypes is advantageous as the phenotypes are already present in a large portion of the population. This is the first correct statement. And the next statement which is correct is statement 4. Notochord in vertebrates is an example of phyletic constraint. So these two are the correct answers to this question. And let us, since we saw that people are getting confused, so I'll explain all the four statements here. Let us go through it one by one and clarify. So the first statement, environmental induction of phenotypes is advantageous. So this is a concept that Professor Subramaniam was referring to, genetic assimilation and accommodation. Environmental induction of phenotype refers to alteration of an organism's physical or behavioral traits in response to changes in its environment. So very simple definition, environmental induction of a phenotype. It means how an environment can induce a phenotype inside an organism. So any phenotype which is conferring an advantage to the survival of the organism, will be induced by the environment, or the organism will acquire that. So organisms' physical and behavioral traits will change in response to environment. This can be advantageous in certain situations because it allows an organism to adapt to different environmental conditions without requiring genetic changes or evolution or multiple generations. So if a certain species now encounters a completely different environment, then such induction of phenotype can be advantageous to that species because it can allow that species to adapt to the different environment. These phenotypes are already present in the population and get selected due to favorable environment. So it's not like any mutation will happen, or genes will change completely, or duplication event will happen, and evolution will happen only after that multiple generations, then we get a new phenotype, so that now the species can adapt to the environment. But many, but the plan in the body, many such traits can be environmental inductible because in a population of the organism there are already certain individuals having that trait. So what happens? Those individuals get selected due to favorable environment. So let's see an example here. I have mentioned an example of butterfly, dev- development and adults. Okay, so we know that butterfly develop from the larva, and then in an adult they grow wings. So in the larval stage, that is just a feeding stage in the cocoon. So those butterflies which lay the larva in warmer season. Let's say some butterflies. So this is a population of all these butterflies. There are some butterflies which can lay the larva, lay eggs, and which will develop into larva in the warmer season, such that in the wet season they will grow into adults. So such butterflies live fast, producing 2-3 generations in a wet season. Conspicuous eye spots deflect predator attention. So such butterflies have eye spots to deflect the predator attention, and they live fast, producing 2-3 generations in a wet season. So they want to reproduce more, and they have certain characteristics such as eye spot to deflect predator attention. Now there is another type of butterfly which lays its egg and larva develops in the cooler season, whereas the adults emerge in the dry season. So such butterflies live long and reproduce towards the end of the dry season, and they have cryptic wing colors against dry foliage to avoid attention. So you see there are two different variants already present in the population. So now what will happen if we take this population into a dry habitat? So eventually we will see the dry variant emerging, getting selected due to favorable environment, because they are already present in the population, and the wet season variant is drying because is dying, and it's getting spotted by the predators. So in such a case, this is the example that we encountered in the lectures also. Environmental induction of flatfish phenotype with both eyes on the same side. So originally the fish had eyes on both the sides. So one eye on the left side, one eye on the right side. But evolution of this flat fish happened very quickly because some population of the this kind of fish was already flat, dwelling at the bottom. So at the bottom you will find more of these fish due to environmental induction, because that environment is fit for such a phenotype, both the eyes on its top to avoid its predators. So similarly, that this is the concept of environmental induction. Favorable environment will favor certain traits and characteristics, and such individuals will be already existing in the population. They will just get selected by natural selection, and that won't take, so changes in body plan would not take much time, mutations and drastic genetic changes. So that's how the first statement is true, because environmental induction is advantageous. The traits or the phenotypes that are already present in a large part of the population. The second statement that we encountered is an example of heterotypy, where insect legs were, so this was the second statement, leg development in insects controlled by UBX, which inhibits distal less gene, ultra bithorax, which inhibits distal less gene in posterior segments through its C terminal polyarginine stretch. So everything in this statement is correct, but this part, polyarginine, is not correct. So what are the mutations? Polyalanine is the mutation. Polyalanine mutation in insector ancestor replication slippage. So what happens? Polyalanine tracts allow UBX to repress distal transcription, distal less transcription. So the phenomenon is actually correct. Ultrabithorax inhibits distal less transcription in the posterior segments of the insect, inhibits abdominal legs, but it is the polyalanine stretch which is responsible for the inhibition, not polyarginine stretch. So this was a technical question, and this is an example of heterotypy. Heterotypy means the change in type of gene or expression. So earlier we had a different kind of ultrabithorax which was not able to repress distal. Now we have a polyalanine stretch in the ultrabithorax which is a different type. So that is heterotypy. Now this kind of ultrabithorax is able to repress distal less transcription, inhibiting abdominal legs in insect. So that's why second statement is not true. It will be polyalanine mutation instead of polyarginine. And the third statement, yellow agouti mice. So if you remember the third statement, pregnant yellow agouti mice, when fed a diet with low methyl donors, will produce dark skinned fit progeny. This is also an example, mostly remembrance type. You just have to learn it by heart. These are certain important examples. Yellow agouti mice. Yellow agouti mice. So the agouti locus governs fur coat color and fat accumulation. So it governs two phenotypes, the fur coat color and fat accumulation. It is a classic example of epiallelism where changes at the epigenetic levels can be inherited. So this agouti mice is a example of epiallelism. We will encounter epiallelism later in the slide where we have a question on epiallelism. So for now, for agouti, changes in DNA methylation leads to phenotypic changes and inheritance pattern. So this is what I meant by epiallelism. Changes in the epigenetic marks like DNA methylation can lead to phenotypic changes. And what is a phenotypic change? So if we feed, so in high methylation, when the agouti locus is highly methylated, it will produce this kind of a mouse. Low fat and dark colored, black brown color mouse. When the agouti locus is low methylation, is epigenetically lowly methylated, then it will form a fat mouse with yellow brown color. So what is observed when a pregnant agouti mother is fed with high methyl diet, it will give agouti brown black pattern with low fat. So this kind of a progeny. So the mother might be yellow agouti, but if you feed the mother high methyl diet, then the progeny will acquire brown black pattern. And if you feed the mother with low methyl diet, it will acquire the yellow pattern, even if it is the black mother. So irrespective of the mother, whatever the diet is fed, the 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 upcoming progeny, the next generation, can have effects, epigenetic changes due to methylation levels increase and decrease, and then this agouti locus will produce a different phenotype. Ajay, you have raised a hand. You can speak. Okay, okay. So the question was the third option was, if a yellow agouti mice is fed with a diet of low methyl donors, it will not produce a dark skinned progeny, because low methyl donors, irrespective of the mother, if you feed a mother with low methyl donors, then the pups will be yellow skinned, because low methylation leads to high fat and yellow brown color. So this is the low methylation phenotype. This is the high methylation phenotype. So that's why this statement is also not true. And coming to the last statement, the notochord is a phyletic constraint. So if we revise our developmental biology class, what is a notochord and how is it a phyletic constraint? Notochord in vertebrates is an example of phyletic constraint. Evolutionary history of certain body plan lays certain constraints which are known as phyletic constraints. So what are phyletic constraints? Evolutionary history of a certain body plan. So, so we'll understand this with a perfect example. Notochord is a transient organ only restricted to embryonic stages, and nevertheless, we are dependent on it at the that developmental time for development of somites and neural crest. So in human embryos, if you see, notochord is not an adult structure. It's not a permanent structure. It's a transient structure. And the presence of notochord is necessary for only at that developmental stage. It is necessary for development of somites and neural crest cells. But if you see the origin of this notochord, it actually started as a mechanism in chordates, which do actually have the notochord in adult stages. So if you go back in evolutionary time, vertebrates arose from chordates. So the first chordates, even if you see now urochordates, they have the notochord even persistent in the adult stages. So in urochordates and in earlier stages of the chordates, earlier chordate organism, notochord is not a transient structure, it is a permanent structure, and it is also responsible for somite and neural crest. The vertebrates which have evolved from the chordates still have the same phyletic constraint even when we do not require the notochord in adult structure. So this is the concept of a phyletic constraint. The vertebrates which has evolved from the chordates is still in a phyletic constraint to use the notochord. Even if it is not present in the adult structure, it will still use the notochord in the embryonic structure to form somites and neural crest. And here is a representation where is the notochord and how does it induce the neural crest cells, also causing the neural tube formation. So this is an example of a phyletic constraint, notochord in vertebrates. Even if it is not present in the adult structures, it is still required in the embryonic structure, which is similar to the earlier ancestor, where it is producing somites and neural crest. I hope this is clear to the students. Let's move on to the third question. Third question is a statement true false question. Statement 1, when excess SHH is synthesized from the precaudal plate, eyes fail to form. And statement 2, SHH from precaudal plate promotes PAC6 expression in the center of neural tube to create a bilateral eye field. So these are the two statements in context of SHH, PAC6 and eye induction from the precaudal plate. Now you guys have to tell me which of the options is correct. Option A, statements 1 and 2 are true and statement 2 is the correct explanation of statement 1. Option B, statement 1 and 2 are true but statement 2 is not the correct explanation of statement 1. And option C, statement 1 is true, statement 2 is false. Option D, statement 1 is false, statement 2 is true. Option C, statements 1 and 2 are false. Now you have to tell me which of the options is correct, whether statement 1 is true and 2 is also true, or either one of them is false, and whether 2 is the correct explanation of statement 1. Let us take some time to accumulate some answers and give the student ample time to go through the questions and come up with the correct answer. I'll reveal the correct answer in 2-3 minutes. Okay, it's time I reveal the correct answer. I see that students have answered option 1. Okay, let me reveal the correct answer. The correct answer is option C. Statement 1 is true, but statement 2 is false. Okay. Now let us understand the development I development in SHH, and what is the role of PAC6 in I field. So PAC6 is a key regulatory gene. It's a transcription factor. Initially, PAC6 transcription factor is expressed in the anterior neural ridge of the neural plate before the neurulation begins. So anterior neural ridge is the frontalmost part of the neural tube even before neurulation has begun. Neurulation means folding of the neural tube. PAC6 establishes a single eye field that later separates into two optic primordia. So PAC6 will establish a single eye field. So this is the eye field here, marked by the PAC6 expression, and SHH expression here. So what does SHH do? Signal for separation of this field is sonic hedgehog expressed in the precaudal plate. So we know now that wherever there is sonic hedgehog, it will inhibit PAC6, creating separation of the eye field. So if you focus on these two diagrams, the red region is the eye field. So the job of PAC6 is to establish a single eye field, but that later separates into two optical primordia by the signaling of SHH. SHH will prevent expression of PAC6 in the middle, creating two different eye field. SHH upregulates PAX2 expression in the optic stalk while down-regulating PAC6, restricting these expression to the optic cup and lens. So this is what I was talking about. SHH will turn on the expression of PAX2, but it will inhibit or down-regulate PAX6, thereby restricting it to the eye field. So this statement, SHH from precaudal plate does not promote PAC6 expression. It does not promote, it actually inhibits PAC6 expression in the center of neural tube to create a bilateral eye field. That is why statement 2 is false. So you guys have to read the question very carefully, because there might be little differences and hints hidden in the question itself. So SHH, so actually PAC6 function here is mentioned very correctly. PAC6 expression will create a bilateral eye field, but SHH inhibits PAC6 and thereby forming a bilateral eye field. So here in the diagram, if we see, for initially there is a region of total, so there is a region of total red here marks the PAC6, but when there is SHH expression, PAC6 is only inhibited in the middle portion, creating two different domains now, which will create two different eyes. So if you remember the Jervine alkaloid and SHH mutation that I was talking about, if there is no SHH, if there is an alkaloid or inhibition of the SHH, then SHH cannot inhibit PAC6, leading to a unified eye field. That is why we have a single eye in SHH mutations or in Jervine alkaloid case. So SHH working fine will produce two eyes due to inhibition of the PAC6. So that is what is the trick here. Now if we quote Professor Subramaniam's slides, SHH separates the eye field by suppressing PAC6 in the center. This is what the statement he mentioned. When SHH is inhibited by Jervine alkaloid, this separation does not happen, leading to cyclopia. So this is what I was trying to explain. And on my right is the molecular diagram showing in the midline SHH will promote expression of PAX2. PAX2 is in the optic stalk. Now PAX2 will inhibit PAX6. PAX2 will inhibit PAX6 in the middle, thereby creating the optic cup. Optic cup, two optic cups on both sides. Now if you do not have PAX2, which is now in turn mediated by SHH, we will have a unified PAC6 expression leading to one single eye. So that is what is the total concept about SHH, PAC6 and division of the eye field. I hope now you are clear. It is clear to the students. So you do not have to worry. I am here to clear your doubts and everything. If you have any questions, you can related to the problems that we are solving, you can ask me. Yes, Ajay, there are two tricky questions. You have to see through the statements and be careful while you're attending the exam, and also to Sakshi and other students. Okay, cool. Let's move on with the next question. So using this image, we have to answer question number 4 and 5. This is an image distribution of showing Darwin finches. So Darwin finches are very famous model organisms which Darwin used to study and to give the theory of evolution. So there are variety of finches. He visited the Galapagos Island to find out different variety of finches with different beak size, different feeding habitats. So using this, we will answer question number 4 and 5. So look at this diagram carefully, and I hope you must have learnt it by heart. Okay, question number 4. Which of the following statements are false with regard to expression patterns in the embryonic beaks of these finches? Which of these following statements are false? Option A, BMP4 levels of large ground finch greater than large cactus finch. Option B, BMP4 levels of large ground finch less than large cactus finch. Option C, CAM levels of cactus fish. Oh, it's not fish is finch. CAM levels of cactus finch greater than vegetarian finch. And option D, CAM levels of cactus fish less than vegetarian finch. Now you have to tell me which of these statements are false with regarding to expression patterns in the embryonic beaks. So there are two players here, BMP4 and CAM, and you have to compare the levels between, so BMP4 between large ground finch and large cactus finch. So if you refer the diagram, large ground finch is this one, large cactus finch. So BMP4 between large ground and large cactus finch, and CAM levels between vegetarian finch and cactus finch. So this is cactus, this is vegetarian. So if you look at the beak size and shape and size and the feeding habitats, you may be able to answer. Also, you have to connect what is the role of BMP and CAM expression. Let's wait for a minute to see some answers. So again, I will emphasize cactus finch, large cactus finch and large ground finch. What is the BMP level? Calmodulin levels between cactus finch and vegetarian finch. This was the question. BMP levels of large ground finch versus cactus finch, large cactus finch. CAM levels of cactus finch versus vegetarian finch. Okay, I see some answers. Sakshi has answered 1 and 4. So you have to spot the false statements. 1 and 4 is Sakshi's answer, and Ajay is answering 2 and 4 are false. Okay, let me reveal what is the correct answer to this question. The first statement which is false is 2. Second statement is false. BMP4 levels of large ground finch less than large cactus finch. This is not correct. The first option is correct. And the next statement which is false, is option 4. Cam levels of cactus fish less than vegetarian finch. It is not true, it is the opposite way. Well done, Ajay, and 1 and 4 are true. Okay. Okay, Sakshi, I think one of your options was correct, so well done to you too. But option 4 is a wrong answer. Option 4 is false. Option 3 is the correct answer. Cam levels of cactus fish greater than vegetarian finch. So let us review ourselves with the levels of BMP and CAM and their role in Darwin finches. If you remember this diagram, so the beak size on the left we have on the A panel, we have beak size. Beak size has three dimensions: length, width, and depth. And the sharp beaked finch, mixed diet of seeds and insects. Low cam, short beak. Low BMP, low beak depth and width. So these are the two players. If we have low calmodulin or low cam expression, we have short beak. And if we have low BMP expression, we have lesser depth or width. So you can then speculate what are the levels in a cactus finch, which looks like this. We have a long beak but lesser depth. So long beak means high cam, elongated beak, lesser depth, low BMP. Probing cactus, flowers and fruit. This is the cactus finch. And for a vegetarian finch, if you see, so in our question we had large ground, large cactus. So let us see BMP levels between large ground and large cactus. We just now learned that BMP level means width of the beak. So if we look at the diagram, large ground and large cactus. So definitely large ground finch has higher depth or width of the beak. This is more wider. So more wider means more BMP. So this guy has more BMP signaling. BMP levels of large ground finch is greater than the cactus finch. This is a true statement. So second statement is a false statement. And cam levels determine the length of the beak between cactus and vegetarian. So cactus, vegetarian. So here you see cactus has a longer beak. So long beak means higher cam and short beak lesser cam. So cactus finch will have higher cam with respect to vegetarian finch. So cam levels of cactus finch greater than vegetarian finch. This is a true statement and 4 is false statement. So 2 and 4 are the false statements, which is the correct answer as I have revealed. I hope it is clear. So you just have to remember what is the function of BMP and what is the function of calmodulin. Then by looking at the diagram, looking at the beak length and the beak width, you can tell easily what is the level of BMP and CAM in that Darwin finch, in that variety of Darwin finch. And depending on that, they have different feeding strategies. Some feed on cactus, flowers, fruit, some feed on insects, some feed on large seeds which need more crushing power. So more crushing power, they need a wide beak but short beak. For a longer beak, they need to probe into flowers. So flowers have nectar, they want to have that and they want to eat the flowers. So they need a longer reach but not so much strength so that they can easily dip into the flower. I think it is clear to the students. Just let me know with a yes or no. We are moving slowly, but I hope that you guys are able to clear your concepts, because then this is the last session and you will have your exams, and I want all of you to do very well and you clear your concepts at least, which is why we are having this live session. Okay, let's move on to the fifth question, which is also related to the Darwin finch. Evolution of finch beaks is mediated by dashed: heterochrony, heterotopy, heterometry, or heterotypy. How can we classify the evolution of finch beaks? Let's take 2 minutes to accumulate some answers and give all the students a chance to think about it. I will reveal the correct answer in 2 minutes. Okay, let me reveal the correct answer. Evolution of finch beaks is mediated by heterochrony and heterometry. So you guys were partially correct. Heterometry, of course, it is heterometry because change in heterometry is change in amount. So if you follow the last terms of these terms, you can easily identify what kind of change it is. Heterochrony is change in time of expression, change in duration of expression. Heterotopy, topo means topology or geography or space. So heterotopy, change in space where it is expressed. Heterotypy, type change, whatever gene or whatever thing was expressing, now that is not expressing, a different type of that is expressing, so that is heterotypy. So that in that way you will be able to visualize and remember it easier. So evolution of Darwin finch beaks is both mediated by heterochrony as well as heterometry. So we just saw an example where we had change in amounts of BMP and calmodulin. So BMP4 and calmodulin represents two targets of natural selection. Together they explain the shape variations in Darwin's finches. BMP4 is regulated heterochronically as well as heterometrically. Calmodulin is regulated heterometrically. So here is a fine line of distinction. BMP4 is a ligand, but calmodulin is a protein. So BMP4 is a ligand which is also regulated heterochronically, means the time of expression of BMP4 also determines its expression. The time of expression as well as the amount of expression of BMP4, both are important. While natural selection will allow certain morphologies to survive the generation of these to survive, the generation of these morphologies depend on variations of developmentally regulatory genes such as those for BMP4 and calmodulin. So calmodulin is regulated heterometrically, mostly heterometrically. The amount of calmodulin present will determine what kind of beak length is going to form. But BMP4, it is regulated in time as well as in amount. I hope it is clear to the students. So many phenomenons, many developmental phenomenons can be explained using uh, all these four phenomenons. So it just says that the toolkit that an organism use for its development is the same. Like the evolution or the creator or any power or whatever mechanism the organism is developing, the toolkit is same. So with the same set of toolkits, it will combine it differently to form different organisms. Whether it will change its time of expression, type of expression, space of expression, amount of expression. So all these can be tweaked and producing newer body plan and organisms. Okay, that was a tricky question again. Let us move on to the next question. Ajay has raised a hand. Ajay, you can speak up. And cal modeling. Okay. Yeah. Yeah, because no, I hmm, okay. I might have to check, but I just found it on the internet. It's not from any book. I don't remember if I can say, but heterometrically and heterochronically, I think uh, it is how it is regulated. It's not from Gilbert. I have it's not from Gilbert. I'm sorry. I have to look. Maybe you are right, or maybe I'm right. I don't know, but I think it's chronically as well because I found it somewhere. Okay, let's move on with the next questions. We are running short on time. I will look at it, Ajay, and if it's not given in Gilbert book, you can write it in the exam. If a question comes, you'll get full marks. So Gilbert is a book that Professor Subramaniam follows, and all the students are also advised to follow. Maybe I had found it somewhere that it's heterochronically as well as heterometrically. Okay, for question number 6, for the model given below that depicts mechanism behind cusp development in molars, identify the graph that best represents relationship between BMP4 and FGF8 over time. Cusp development in molars, so teeth development. This is the relation here, BMP and FGF8. Epithelial proliferation, mesenchyme proliferation, and how they are regulating it, and what is the relationship between BMP and FGF8. If we depict it in a graph, then how it will be. So these are your options. On the y-axis we have relative concentrations, and on the x-axis we have position. The red line correspond to BMP4 and blue line to FGF8. And you have to tell me what is the correct representation showing the relationship between BMP4 and FGF8 in cusp formation. Yes, yes, you can see here. I couldn't accommodate the options in a single slide because the options were all graphs, and they would not be clearly visible if I put the options with the question. So I made a separate slide for all the options. So if you remember, yeah, yeah, once you guys register this image, or if you remember the molar development or the dental development, and what is the role of BMP and FGF signaling. So again, we have two different signaling and representing the cusps. Cusp development is the depressions in the teeth of the molar, and then we have those mountains and cusps. So our molars are made for grinding. So they'll have some depressions and they'll have some elevated surfaces. So in those, what is the role of BMP and FGF8 and how can we represent them? So these are the options. Red line is for the BMP4 and blue line is for the FGF8. Okay, I see Ajay has answered option 3. Yes. And Sakshi, no idea, sir. It's okay. Okay. Let me reveal what is the correct answer to this question. Ajay is very correct. Well done. Option C is the correct answer. BMP has two mounts is correct. The representation you were mentioning too. The pink lines, BMP expression. Then we have FGF expression, FGF8. So if you look at this, then the cusp that will form will be in this area where there is low BMP and medium FGF. So this is based on a model which we popularly is known as Turing pattern formation model. So this you can find in the Gilbert book, and also if you skim through the lectures. Turing pattern formation refers to a type of pattern formation in biological systems that can occur through interaction of two or more morphogens that diffuse through tissues and react with each other. So during the morphogen constraint, Professor Subramaniam has mentioned this, and this is also known as Turing pattern formation. Two different opposing morphogens can interact and diffuse through the tissues, react with each other to form different structures. So we have activator P, which stimulates production of inhibitor S, rapidly diffusing inhibitor S. S diffuses quickly and inhibits autocatalysis of P, and slowly diffusing activator P. So this is what we saw in the graph earlier. So the green one is the BMP, which stimulates production of inhibitor, and the red ones are the FGF. So if we compare the Turing model, relative concentration, so at a time when there is inhibitor at a constant level, there is activator. Then slowly, with regard to position, the activator concentration will peak at certain levels where there is, and it will coincide with the peak of the inhibitor. But since the inhibitor diffuses very quickly and inhibits autocatalysis of P, there will be some amount of inhibitor still there when the activator levels are still low. So the activator, slowly diffusing activator, will not be able to autocatalyze or stimulate more inhibitor. Then in the next phase, when the activator slowly rises up due to its slow diffusion, then we can again see production of the inhibitor. So this is the mechanism how BMP and FGF play to make molars. So let us learn it in the molar context. BMP is a morphogen that plays a crucial role in tooth development. It is involved in specifying the shape and differentiation of tooth structures, including cusps. BMP signaling promotes formation of dental tissues. And FGF signaling is another important signaling involved in regulation of proliferation and tissue differentiation. They play a role in shaping the overall structure of the tooth. In the development of molar teeth, there is a dynamic interaction between BMP and FGF. Both pathways can influence differential of dental cells, and combined effect help to establish complex pattern of cusps, which are elevated structure on the surface of molars. So these elevated structures and depressions that I was talking about are present on the surface of molar. And how it fits into Turing pattern formation? In context of molar development, BMP can act as an activator. So this is what I was talking about. The green line here represents the activator, promoting certain cell differentiation, while FGF can act as inhibitor, restricting these processes in certain areas. So this inhibitor will only restrict the cell proliferation in these peaks where the BMP is active. That's why we get these elevations. the elevations in the molar surface. The interaction between BMP and FGF gradient can lead to emergence of patterns such as arrangement of cusps and grooves on the molar teeth. So this is how BMP fits as an activator and FGF as an inhibitor in the Turing pattern generation model. Okay, quickly moving on to the 7th question and 8th question. So this is a question uh, where the statement is given above. Using this information we will solve question 7 and question 8. Assume that a flower has three color alleles W, P and R. It is usually grown at temperatures ranging between 20 to 25 degrees Celsius. Okay, there is a flower which has three color alleles W, P and R, usually grown at temperature 20 to 25. In absence of a heat shock, all alleles remain white. So when there is no heat shock, it means normal temperature growth, 20 to 25 growth, all the three alleles W, P, R will give white flowers. When subjected to a heat shock, the ones with W allele continue to produce white flowers. So when temperature is raised, W will produce white flowers. The ones with P allele produce pink flowers. P allele will produce pink flowers and ones with R allele produce red flowers. So W will remain white, P will produce pink and R will produce red flowers when the heat shock is given or when these flowers are grown at higher temperature. The plants that continued to produce white flowers post heat shock were selectively bred for 15 generations. Okay, the plants that were continuously producing white flowers even after heat shock. That means the plant with W allele, okay, is bred for 15 generations. So you understood the question? Now the question is, which of the following statements are not true or are false about 16th generation of offspring? So the plant that was propagated for 15 generations, now in the 16th generation, what will hold true and what is false? So the question is, which of the following statements are not true? Option 1, at 22 degrees Celsius. 1 and 3? Okay. Oh, so you have encountered this question earlier. Okay, so okay, okay. And Sakshi, will you be able to answer this flowering question? So the plant with W allele, when bred for 16 generations, which of the statements will be false, which is not true? Okay, 1 and 3. Since you guys have already encountered it, you answered it very quickly. Very good. So the correct answer, let me reveal them for all other students. Option 1, at 22 degrees Celsius, all of them have red flowers. This is not a true statement. And option 3, when subjected to heat shock, they produce pink or red flowers. So Sakshi and Ajay both are really correct. Very good. Let us understand a little about this arrangement. So in our question we had plants with three alleles, color alleles, W allele, P allele and R allele. So W allele in 22 degrees Celsius as well as over 22 degrees Celsius, 25 degrees Celsius, it will always produce white flowers. P allele at 22 will produce white flowers, but greater than 25 it will produce pink flowers. And R allele 22 white flowers, greater than 25 red flowers. So if you take the plant which is constantly producing white flowers even after heat shock, which is the W allele plant, we breed it for 16 generations, then we do not have these alleles. P and R allele are not there. They are lost. It's just the W allele. So now we will have a plant similar to the phenotype of W allele. So at 22 degrees Celsius, they will have white flowers and they are no longer sensitive to heat shock and flowers will remain white. These two are the correct statements. Okay. And question number 8, which statements are correct about what is happening in the above scenario? So if you try to explain the above scenario, which of the statements are correct? Option A, environment both induces and selects for a phenotype. Option B, this process can only happen in artificial conditions. Option C, environmentally induced phenotype can never exist in absence of initial stimulus that gave rise to them. And option D, it is an example of genetic assimilation. Which of the statements are correct? And I see Ajay has answered 1 and 4 and Sakshi also 1 and 4. Given that you have already accounted for this question, so we'll move through quickly. Statement 1 is correct. Environment both induces and selects for a phenotype in this scenario, and statement 4 is also correct. It is an example of genetic assimilation. Well done. Well done, Sakshi and Ajay and other students. Okay. So if you learn about a little bit about genetic assimilation, here is an example. What is genetic assimilation? It describes a process by which environmentally induced traits or characteristics that were initially acquired during an organism's lifetime become fixed in the organism's genetic code and are passed on to subsequent generations. So here we saw in the flower example, so that trait, those alleles of the color trait, W, P, R, those alleles were already existing in the population. But via genetic assimilation, when they were bred for 16 generations, via genetic assimilation, those traits induced, environmentally induced traits, now the environment, the heat shock environment is really conducive for the white alleles to thrive. So environmentally induced traits got assimilated and became fixed in the organism's genetic code. So now the white allele is fixed in the organism's genetic code, which is environmentally induced, which is conferring some advantage to the plant, which is in presence of heat shock also there will be white flowers. So this is an example of genetic assimilation. Another example on the right, population of island tiger snakes. So here is a population of island tiger snakes with the normal ones, which became, so the island ones got separated physically, started living in the island, have become isolated on islands and have larger heads to cope with large prey animals. So on islands we have larger prey animals and lesser diversity of prey. So these snakes have developed larger heads. So how? There were certain variations already existing in the snake population with a larger head, and they got selected when they move into the island. Young populations have larger heads by phenotypic plasticity, whereas large heads have become genetically assimilated in older populations. So large heads in the older population have become genetically assimilated. So here is an example of genetic assimilation in which environmentally induced characteristics can be fixated in an organism and become the part of the phenotype of an organism. Okay, you have answered all the questions correctly now. Okay, let us learn how genetic assimilation works. So initial environmental induction, an individual organism within a population exhibits a phenotypic response to environmental conditions. So some individuals in a population show some response against the environment. This response may be beneficial for its survival and reproduction. So let's say that response is beneficial for its reproduction. However, this trait is not initially encoded in its genetic makeup, but is instead a result of environmental factors. Then we then then a process of selection happens. Individual with that induced trait have survival or reproductive advantage over those without it in their current environments. So what will happen, these individuals will thrive, will reproduce more and give rise to the next generation. As a result, they are more likely to pass on their genes to the next generation, potentially due to increased fitness. Then there is heritability increase over time due to selective advantage conferred by the environmentally induced trait. Genetic changes may occur. These genetic changes increase the likelihood that offspring will also express the trait even in the absence of original environmental conditions. And as the trait becomes more strongly associated with the genotype, it becomes genetically assimilated. And fixation through successive generations, the environmentally induced trait becomes fixed in the population's gene pool, and population no longer relies on environmental cues to express the trait. So this is how genetic assimilation is observed in a population, where environmentally induced traits can be fixated in certain individuals, which confer higher fitness to the offsprings. Okay, moving on to the ninth question. Which of the following are correctly matched? Option A, development of bat wings, heterotypy. Option B, loss of snake limbs, heterochrony. Option C, loss of eyes in cave fish, heterometry. And option D, enhanced uterine prolactin expression, heterotopy. Yeah, so which of the following are correctly matched? Development of bat wings, heterotypy, loss of snake limbs, heterochrony, loss of eyes in cave fish, heterometry, enhanced uterine prolactin expression, heterotopy. And Ajay and Sakshi have mentioned option 2 and 3, which are the correct answer. So, loss of eyes in cave fish is heterometry, correctly matched. Loss of snake limbs, heterochrony, is also correctly matched. So let us learn a bit about some of these options. Batwing development. In batwing development, it is not heterotypy, rather heterotopy, because a new player comes into expression. FGF expresses in the interdigital mesenchyme, inhibiting BMP regulated cell death. Thereby, there is no cell death in the bat wing, and we have the interdigital mesenchyme forming the cartilage. That's how bat wing develop. So it is heterotopy, change in type of gene expression. So FGFs come into the picture, not heterotypy. Sorry, heterotopy is the my bad. Heterotopy is the change in region of expression or geography of expression, spatial change. So FGFs come into picture in the space, and then they inhibit the BMP cell death. Fish, cave dwelling fish eye development is correctly matched, is an example of heterometry. Heterometry is the change in amount of gene expression. Increased amount of SHH enlarges the area of PAC6 absence, preventing eye formation. So when we learned about the SHH inhibiting PAC6, PAC6 which forms the future eye. So there is more amount of SHH in the cave dwelling fish, which prevents eye formation. However, increased amount of SHH enlarges the jaw and increases number of taste buds, useful adaptation for the cave fish. So these are certain adaptations which are example of heterometry. And the loss of snake limbs is heterochrony, loss of snake limbs rather than formation of ribs. So the snake has ribs all over its body due to SHH expression in the hind limb. So that is also correct match of heterochrony, loss of snake limbs, and loss of eyes in cave fish, heterometry. Okay, and the last question of this session, epialleles such as those responsible for Peloria mutant in toad flax. Epialleles such as those responsible for Peloria mutant in toad flax are dashed, are sequence variants, differ in chromatin structure, cannot be inherited across generations, both B and C, None of the above. Let's wait for a minute to collect some answers. I can see that Ajay and Sakshi have both answered option 2. Oh, it was also a practice question. Okay, let me reveal the correct answer. Epialleles differ in chromatin structure and they are not sequence variants. They can be inherited across generations. So all these characteristics define epialleles. Ajay and Sakshi have answered both correctly. Well done. Good job. So if you see, epialleles have same DNA sequence but different epigenetic landscape. Changes in such as changes in chromatin organization and structure, methylation marks, acetylation signatures, etc. So the same gene might have different epigenetic landscape. So here we see some parts of the gene are accessible for transcription. So it transcribes a different amount and time and kind of gene, whereas these are heterochromatinized or more methylation marks which suppresses its transcription. Thereby we generate different kinds of coat color. So this is an example of apiallelic gene, and we also learned about this in the Agouti question. When the Agouti locus is actually apiallelic, without change in DNA sequence, it can resemble, it can bring about changes in the methylation mark, controlling fat locus, fat accumulation as well as coat color. So you guys are very correct. Okay, with this we come to the end of our practice problem session. Let me stop presenting.
Some question first. Okay, please.
Oh, you want to see question first? Okay, I'll have to present it again. Yes, so this was the question number 1. Match the following.
So Germain, I know that's okay. Then
Yeah.
Developmental is okay. Then Buchnera is a symbiotic gene. So it's along with E means IPBK means that is with that pea plant,
Pea plant.
right?
I thought
It's
Buchnera
pea
is an
plant.
aphid.
And fourth was A corn
Heterotypy. Corn gloom. Yes.
kind kernel is expressed, okay. And fifth phylogenetic stage is parenchyma P. Okay, thank you, sir.
Yes. Okay. Thank you. I'll stop the
And
presentation with this.
sir,
Yes. Any other question
we
you have?
had that zona pellucida problem. I
Yeah.
think I this even you and me both were correct. So it will its main function is to prevent implantation in fact.
So if zona pellucida is absent, then it can be implanted.
Absent if it is broken, is prematurely if it is broken, the
Yes.
embryo gets implanted in the tube. That you
In the fallopian tube also. I think I remember the question. If there is a uh, zona pellucida mutation or something, then can it implant in the fallopian tube? It was the question, I think.
it is given on page number 304, 19
Okay.
specific.
So it
One
can.
word.
So have you gone through it? Can it really
Yes,
implant in the fallopian tube?
yes, yes, sir. If you want, I
Okay.
can
Okay.
give.
Okay. Okay. Good. Good. Great. Okay.
Second
With that,
question is about that Darwin's tree.
yes.
Something is mentioned that calcium eating or some calcium ion. I didn't understand that.
Calcium ion is actually related to calmodulin. So calmodulin is a calcium binding protein. So calmodulin, the expression of calmodulin or the activity of the calmodulin will be affected by the amount of calcium bind to it. So active calmodulin will bind calcium and then perform its function, but calmodulin not bound by calcium will be inactive. So that's where calcium ion comes into picture. So calmodulin is responsible for long weeks that we learned. Anyways, you answered that question correctly.
Involving there will be something hard. That's why excess calcium is secreted. For that, their being gets short. So, no role of PMP and calmodulin basically. That's what they are trying to say.
Yeah, yeah, yeah. Yeah, you can say that because since beak is a bony structure, so excess calcium will actually
Help them.
harden the beak.
Sequestering only. That's
Because mineral deposits will be more on the beak and it will generate much force. So apart from thickness, which is conferred by BMP, then thicker beaks will have higher, if there is a higher calcium deposition, then they'll have harder beaks.
and last thing, sir, Turing model. I tried to read it two times, three times, but
Yeah.
still that dates and curves I'm not getting.
So
Mice and
it's
hope.
just saying that the inhibitor or here FGF8 is restricting
That
the
chart
expression
I
of
got,
BMP.
but what with the help of chart when they are explaining with the prediction of that mice and more, you predict that and you get that, observe that.
Yeah.
That's what. Please, if you can explain.
So Turing model just as I said, so basics is what I can tell you. The inhibitor, which is FGF8, and the activator, which is BMP. Inhibitor is fast diffusing, means or it will move faster than BMP. So FGF8 is faster than BMP, means if you put FGF8 and BMP in a cell, FGF8 will cover most of the cell in lesser time than BMP. So what it does, only where it restricts the domain of BMP expression, that is what FGF8 does. So if you see, that's where the cusps are forming, or that's where the elevations of the tooth are forming. So wherever there is high BMP expression, there is elevation of the tooth. Now this BMP can be, if it's allowed to slow diffusion, then it can form all over the teeth and then the teeth will just have elevations. There is no depression. So to cause that depression, the FGF is restricting the activity of BMP in a small area. That's where we have the that's where we have the cusp, molar cusp. And now after a certain concentration, so the inhibitor concentration does not increase beyond a certain limit, does not fall down beyond a certain limit. So after a certain time point, the BMP will still start diffusing and form another peak. So slowly the BMP will form another peak, and such peaks will be observed. So that's why we had two BMP crests and then lower FGF.
That's easy. That would have been easy in geography because we get alternate dark and white bones. But then it's explained with different teeth because of the probability of fossils, right? So
Yeah,
I think
whichever is
yeah.
easier for you guys to understand. So I think your exams will be on around 30th of this
No,
month,
29th October.
29th October. Okay.
And thank you, sir. And sorry if by mistake we had lured you or misbehaved
No, 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 no.
in the end because
This is
most of the time I was with you.
it's fine. These sessions are for discussion and for arguments. So any of us can be correct, can be wrong, and I'm not a see. I'm a teaching assistant. I'm a PhD student. So just like you, so we all both can make mistakes, and it's completely fine. We are here to find out the right answer and clear our concepts about the course. So that's what NPTL is expecting us to do. So it's fine, and all the best for your exams. With this, I'll end your session. I think there is an extra session that NPTL had emailed me that there can be an extra session on Monday, uh, which I had filled for at 11 a.m. maybe. I'll prepare some questions from here and there in NPTEL question banks, and then
Sir,
see.
I'm not available till on Saturday. It's fine. I can availability at 3 p.m. On last Saturday, I was in meeting in on CVC conference, and still I was with you.
Yeah, yeah,
But
yeah.
better if
Yeah,
you
I know,
like.
Ajay, but due to due to other
Because
responsibilities, I
PTL people, they are working people most of the time.
yeah,
But if they take in the morning 7 a.m., it's okay. But 11 a.m. office hours, it's very
I know, but I cannot help it. Those were the times that I was free to take the class. I have to prepare also. So after this class, I have to make slides for the Monday
already
class.
two, three meetings are lined up. So it's
Yeah,
very difficult.
it's okay. The
Thank you.
session will be recorded, and you can visit YouTube and watch anytime if you want to clarify. The slides have uploaded, will be uploaded, and you will get access. And you are already following some of the lectures as well as books. So I don't think you will have any problem. You are doing very great.
I think so there is a much of a difference between seeing a recorded session and live session.
Yeah, yeah, yeah. That's why NPTEL had asked us PMRFs to take classes to help students with this course. But unfortunately, very less
Master is
students
not
have
different.
registered for the exam, as you said, for developmental biology.
Okay. Thank you, sir.
Thank you, Ajay, and all the
I
best.
will try to join
Yes, yes.
on Monday.
Okay,
Thank you.
okay. Thank you. I'll end this call.
Thank
Bye bye.
Actually, very thank you. Thank you for all